And just to introduce Quirin, he received his PhD from Humboldt University in Berlin and the German Rheumatism Research Center in Berlin. His PhD work focused on host-pathogen interactions between NK cells and human cytomegalovirus. And he's currently working at Karolinska in the lab of Karl-Johan Malmberg, where he studies antiviral responses of NK cells and aims to develop next-generation NK cell immunotherapies against leukemia. So thank you very much, Quirin. Over to you.
Thank you so much for the introduction, and I'm also really happy to having received the invitation to actually give a presentation at this meeting today. You mentioned correctly, right now I'm working in the lab of Karl-Johan Malmberg at the Karolinska Institute, but I think the majority of the data that I will be showing today is from my Berlin time when I was working at the German Rheumatism Research Center. Today, we've heard actually a lot about different immune cells, not really much about natural killer cells. That's why I would start kind of slow with like a very detailed introduction, but then I'll try to pick up more speed as we go along. So NK cells have been initially identified because of their capacity to naturally kill leukemia cells. And what this video will hopefully show you in a second, it's from a collaboration partner, is that NK cells are really, really good at killing leukemia cells. So in this video, there's this one red cancer cell, green cancer cell, sorry. And the bright green color indicates that the cell is alive. And together with this cancer cell, there are some NK cells in red in this microwell. And after a short contact, the NK cell kills the tumor cell, which is then indicated by the loss of this bright green fluorescence. So NK cells can really quickly perform cytotoxic functions and kill cancer cells, but they can also secrete soluble factors such as chemokines or cytokines. And that we know from the previous talk as well, that cytokines such as interferon gamma or tumor necrosis factor can be very important for crosstalk and to kind of coordinate immune responses. These functions of NK cells, they are controlled by really a broad set of receptors. NK cell receptors are germline encoded, so they're kind of stuck with the specificity. They cannot change their specificity. But to kind of compensate for that, NK cells express really numerous receptors, and receptors can be either activating or inhibitory. Activating receptors mainly bind to ligands that are induced upon cellular stress or by a pathogen infection. And the majority of inhibitory receptors actually recognizes HLA class 1 molecules as self-ligands. And so if an NK cell encounters a host cell that displays kind of increased or aberrant expression of ligands for activating receptors, then that leads to activation of the NK cells so that NK cells release their cytotoxic payload and also their cytokines and chemokines. So that NK cells do not eliminate every cell that is maybe stressed just during proliferation. And there's a counterbalance mechanism. When this host cell that is encountered expresses sufficient levels of self-ligands and the NK cell expresses the kind of cognate inhibitory receptor, then that this inhibition that is received through that interaction overrides the activation, and this cell is left alone. And then in a very special circumstance where we have, for example, a virally infected cells where also self-ligands are downregulated in concert with an upregulation of activating ligands, this kind of really pushes the balance of NK cells that express the right combination of activating receptors and then of inhibitory receptors that detect the absence of a self-ligand, and that then leads to a very strong, very robust activation, which is called missing self-recognition. So I think this is important to keep in the back of our heads how this kind of activation of NK cell happens on a cellular basis. But for this talk, I will mainly focus on the activation side of things. And I think one important question is what is the contribution actually of these natural killer cells to host defense? And there we have learned a lot from rare genetic mutations, because patients that harbor genetic mutations which lead to NK cell deficiencies, these patients have actually quite clear phenotypes because they suffer from really severe and disseminated virus infections, mainly caused by herpes viruses, such as varicella zoster virus, Epstein-Barr virus, or also human cytomegalovirus. And within the kind of NK cell immunology field, I think especially cytomegalovirus is a very, very interesting pathogen, because during an acute infection, it has been shown that a very specific subset of these natural killer cells really go into kind of proliferative overdrive and expand. And this population that responds during CMV infection expresses one activating receptor, or mainly one activating receptor, which is called NKG2C. In line with that, even so, if you have cleared the infection and CMV is, I mean, it turns into the latent phase. But if you look at healthy donors and stratify healthy donors for having anti-CMV antibodies, so for CMV seropositivity, then also in these healthy donors, you find in CMV seropositive donors increased frequencies of this subset that has been expanding during acute infection. And in addition to that, this particular subset has kind of changed its phenotype. So if you imagine this here as the NK cell repertoire, where you can find different subsets, then you will find a small frequency of NK cells that expresses this activating receptor in CMV seronegative donors, healthy blood donors. And if you look into CMV seropositive donors, this subset is really increased in frequency and has also changed its phenotype here, indicated by maybe a change from blue to red. This phenotypic change can include a higher expression of some receptors, lower expression of other receptors, and also includes epigenetic changes. And because these data kind of indicate that this particular subset undergoes adaptation in response to CMV infection, they are referred to as adaptive NK cells. I fully understand that this is maybe not a correct term to use here, but it's right now what the field has kind of landed on, and I'll use it in quotation marks. But what I think is, in addition to that, of interest is that this is not an yes or no response. So these adaptive NK cells, they are never present in CMV seronegative donors, but they are not present in all CMV seropositive donors, really suggesting that there seems to be some kind of variability or heterogeneity in this system. So this really caught our interest. And luckily for us, when we became interested and we started the project, it was already clear what the ligand of this activating receptor is. That has been described. The ligand for NKG2C is HLA-E. Similar to other HLA class 1 molecules, HLA-E requires peptides to be really stabilized, for stably expressed at the cell surface. And another, I think, key observation that had been made was that cytomegalovirus, one gene of cytomegalovirus called UL40, actually can encode peptides which can bind to HLA-E and stabilize it at the cell surface of infected cells. So when we started this project, we really thought that NKG2C could be a kind of potential recognition molecule for CMV-derived peptides during CMV infection. And we tried to ask the question, or we tried to answer the question, whether CMV-derived peptides presented on HLA-E could be the ligands that drive NK cell responses through NKG2C. Plus, we were also interested whether these CMV-derived peptides could kind of contribute to the heterogeneity that was described in the response. To initiate the project, we first really analyzed the peptide repertoire that can be encoded by cytomegalovirus. For that, we did a kind of integrated analysis of many sequences that were already available in databanks. And what we also did in parallel, we isolated CMV DNA from patients that had an acute ongoing CMV infection. You can detect CMV DNA in the blood, and then you can amplify the UL40 sequence and really determine which peptide is encoded by this specific virus strain. And you see here the results of this analysis. So one thing is clear, that 3 major peptide sequences really make up almost 2/3 of these more than 200 clinical isolates. But then there was also lots of heterogeneity in kind of the last third, where we found some peptide sequences that were only found in 2 or even only 1 of the investigated isolates. And on the right-hand side, you can also appreciate in this kind of conservation logo that especially position 8 of these non-numeric peptides seem to be a hotspot for mutations when looking at these clinical isolates. And now to test kind of the functional impact of these peptides when they are presented on HLA-E and NK cells encounter them, we used a very, very reductionist model. So we opted for a murine RMAS cell line that has been transfected with HLA-E and human beta-2M. But RMAS cells are top deficient, so they cannot present intrinsic peptides at steady state. But when you add extrinsic synthetic peptides, then and these peptides bind HLA-E or stabilize HLA-E, then you can detect actually surface expression of HLA-E on these mouse cells. And as a next step, we then took isolated NK cells from CMV seropositive donors, where we knew that this NKG2C positive population had been expanded in vivo. So to co-culture them and read out what the functional consequence of the peptide HLA-E complexes are for NK cells. And one marker that we use for these readouts is CD107. That's kind of a marker for degranulation and can be used as a surrogate for cytotoxicity. And what you can see here, first of all, if you co-culture human NK cells that express NKG2C with this mouse cell line without peptide, there's no activation because these cells can really not detect, or there's no ligand on the cells that would activate these, the human NK cells. And then if we go through some of these peptides candidates, so there was one peptide that was really, really rare within the clinical isolates and had actually mutations, not in the 8th, but in 2 different positions. And that also did not lead to any activation at all. And then other peptides that were more common, such as this variant and this variant with the isoleucine in position 8, that induced kind of robust activation. And we found the strongest activation of these NKG2C positive NK cells in also a very rare peptide that had a phenylalanine in position 8. So this really hierarchy of peptides of no activation, intermediate activation, and high activation was observed in several donors and also observed for several other functions, such as chemokine expression, TNF, and interferon gamma as cytokines, really suggesting that variations in the sequence of these peptides from CMV UL40 can have impacts and really control the activation of NKG2C positive NK cells. We observed this peptide hierarchy also over a broad range of peptide concentrations. Again, this peptide variant here in gray with the two mutations in position 5 and 6 that did not lead to any activation at any concentration that we tested. And then this very, very strongly activating peptide, this higher activation was conserved also throughout all concentrations tested. And I think one important point here is that this is really not correlating to HLA-E surface stabilization on the target cells. Because if you look, for example, here at 10 micromolar, you can see that the peptides here represented by blue dots induced almost twice as high HLA-E stabilization, but led to lower activation compared to the phenylalanine containing variant, really suggesting that it is not a function of the quantity of HLA-E that is stabilized at the cell surface, but it could really be kind of a qualitative aspect of the HLA-E peptide complex. So until this point, we had kind of a correlation of CMV DNA sequences and these synthetic peptides. But we were really, really interested whether such a peptide recognition could also actually happen during an actual infection. And for that, we teamed up with Martin Messele and his group, who are excellent CMV virologists. And what they did, they used TB40E, also a strain of CMV, as a template and kind of reintroduced important immunoevasion genes into this CMV strain to make the strain as close to clinical isolates as possible and not work within kind of a lab artifact. And after that, they generated 3 different mutations in the UL40 sequence without touching the remaining genome, so that we had 3 very controlled viable CMV strains that only differed in this UL40 peptide sequence. And UL40 does not really play a role in infectivity, And in line with that, we found that these 3 strains led to very similar infection rates in primary human endothelial cells. So the next step, we added to these infected cells, we added again human NK cells, where we knew there was a reasonably large NKG2C positive population present, and analyzed the activation of these cells. And also here in this model of an infection in vitro, you can see that the peptide hierarchy is conserved with this variant at the phenylalanine inducing the highest activation, the isoleucine, kind of an intermediate weak activation, and this other rare variant, the lowest activation. If we look in the same assay in the same well at NK cells that do not express NKG2C, so they are NKG2C negative, we could not really find consistent differences between the endothelial cells that were infected with different strains, really suggesting that the peptides are differentially recognized by NKG2C, positive NK cells, and that NKG2C really equips NK cells with this capacity to differentiate between CMV strains based only on the UL40 peptide sequence. So since our data imply this kind of peptide-specific recognition, we became interested whether that could also not only contribute to activation, kind of short-term assays, but whether these UL40-derived peptides could also be involved in generating these adaptive NKG2C positive NK cells and expanding them. So to test that, we changed basically the source of our NK cells, and we stepped to CMV seronegative donors, where this NKG2C positive population is present, but usually at quite low frequencies, and they do not have this differentiated phenotype. And we co-cultured NK cells from these donors with, again, peptide pulse target cells, and in the presence of pro-inflammatory cytokines IL-12 or 18, because we know that these 2 cytokines are really induced in vivo also during an acute CMV infection. And then what you can see here is that both at the frequency and in the absolute number, again, the peptide sequence, the peptide hierarchy was conserved with this sequence not really leading to an increase, and then this intermediate sequence really inducing increases in frequency and absolute numbers with actually a lot of variation, and the peptide sequence with the phenylalanine again leading to the highest increase both in frequency and absolute numbers, suggesting that the peptides together with pro-inflammatory cytokines could contribute to the expansion and proliferation of this subset of NK cells. I mentioned that here before, and I put it back up again just to remember some of these markers, because these cells do not only expand when they encounter CMV in vivo, but they also change their phenotype. So we also compared that in the same co-culture system, and we treated again NK cells without or with pro-inflammatory cytokines, and then with this peptide that does not really activate NKG2C positive cells, and this peptide that leads to a very, very strong activation to basically have the 2 extremes of the situation. And there you can see that especially the combination of the cytokines plus very, very strong activation through NKG2C led to the highest increases of CD2 as described for CMV seropositive donors. It led to the strongest decrease in cyclic 7 expression, also kind of paralleling what has been shown ex vivo for these donors, and also an increase in educating KIR and several other protein markers. In addition to that, I think one hallmark of these adaptive NKG2C positive cells from CMV seropositive donors is that they also undergo epigenetic changes. And I think one key change here is in the interferon gamma locus, where an enhancer that is called CNS1 for conserved non-coding sequence 1 is demethylated in this population and actually allows the population to produce more interferon gamma. And when we check that in our kind of co-culture model, we also observed that both the peptide alone and cytokines without a strong activating peptide could induce some degree of demethylation, but again, the combination of pro-inflammatory cytokines and strong activation through HLA-E peptide complexes led to the most pronounced loss of methylation at this particular locus. So taken together, these two slides, basically, or the data on these two slides, suggests that the combination of cytokines and HLA-E peptide complexes can really guide the differentiation of NK cells from CMV0 negative donors into this adaptive population as it is observed in CMV0 positive donors ex vivo. And to substantiate our in vitro data, we followed a cohort of allo hematopoietic stem cell transplant recipients. We were especially interested in two groups. One group that was CMV0 negative and did not experience reactivation, kind of as a double negative control. And then some of these patients, because of the immune suppression, reactivate CMV. And during CMV reactivation, we again isolated DNA, CMV DNA from the plasma and sequenced the UL40 region to determine the peptide sequence of the virus that is at this moment causing reactivation. Then we stratified for the virus sequences over these peptide sequences and phenotyped them, the NK cells. We looked both at the frequency of NKG2C and kind of a differentiation looking at different markers. And what you can see here is that we saw kind of maybe a trend, but this rare peptide is really, really rare, this strongly activating peptide. But then especially for differentiation, we could observe that it was more pronounced when in patients that had been experiencing a reactivation with a virus that encoded for this highly activating peptide. Again, this was only the case in NK cells that have the matching activating receptor and not in NK cells that do not express NKG2C, suggesting that also in vivo, these peptides could contribute to shape the phenotype and the differentiation of these cells. So to sum that up, I think what we try to show in that in this set of experiments is that, first of all, CMV can really provide peptides presented on HLA-E, and then these peptides are recognized by NK cells that express NKG2C, and that can lead to different degrees of activation. And now, especially in combination with pro-inflammatory cytokines that, for example, could be produced by dendritic cells or other myeloid cells during acute infection, the interaction can now lead to different outcomes. So if there's no interaction, these cells do not really expand and they do not really differentiate much. Whereas if there's a very, very strong interaction, that will likely result in a very good expansion and a high proliferation, plus also in kind of very pronounced differentiation into this adaptive phenotype. Whereas when you have a weak interaction, which is actually quite common in the clinical isolates that we looked at, then you can end up with different outcomes, probably determined by other factors such as co-stimulation, duration of the infection, or also degree and severity of the infection. And then we propose that these peptides can be one factor that contributes to these variable pools and to this heterogeneity within CMV0 positive donors, kind of that it's not only about being CMV seropositive or negative, but it could be an additional layer of by which virus you have been infected that has caused your seroconversion. And this is now my last slide, basically just a teaser, because I think one very justified question is cytomegalovirus the only virus that can provide HLA-E stabilizing peptide? And I think in the last couple of months, we've identified another virus that can definitely do that. So it looks like the SARS-CoV-2 genome can also contain a peptide that can bind HLA-E here, that is HLA-E at the surface of after treatment with synthetic peptides. And we've tested several candidates of SARS-CoV-2 peptides, but one is really seems to be a very, very good stabilizer. And we're currently investigating the functional responses. And the preliminary data suggests that it could be that this peptide is involved in activating NK cells. But we will hopefully have soon more data to show on that. And with this, I would really like to thank actually a large group of people from the Berlin side, especially Chiara Romagnani, who was really, really heading the project, and Timo, who contributed a lot as well. And now at Karolinska Institute, I'm also integrated into a really amazing network of national and international collaborations and scientists. And I would really like to thank my mentor, Karl-Johan Malmberg, for all of his support. And I thank all of you for the attention and for sticking with me until the very, very last talk here.
Great, thank you very much indeed, Quirin. That was really interesting. So obviously we'd like to open the floor for questions. And as usual, if you have a question, please just press the raise hand button, or you can unmute and ask. I'd like to kick off, and as the precedent was already set. So Quirin, one question I had, and it may be really, really simple in the way I'm looking at it, but so we make HLA-E pentamers for tracking antigen-specific cells, and obviously these bind to NK cells. So the epitopes that we have are these very same sequences, the VMAP sequences with all those different variants that you've described. And in my understanding from before, we'd spoken, I just saw those as HLA-C leader sequence derived peptides. And obviously, and you sort of alluded to it in your last slide there about viruses presumably hijacking that self-peptide look to stabilize the complexes. That's exactly what viruses like to do. And so I just wanted to, I was just going back to thinking, well, from your experiments where you're looking at these peptides, do you know for sure, obviously there was the RMAS cells where you were just using synthetic peptides, but do you know those peptides were actually from the CMV UL40, or was it potentially from HLA-C? Because I guess the same sequence you don't necessarily know.
So like that is an excellent question. I think it's also all true. So CMV, maybe I can show it on this slide. So I think CMV really, because for CMV, it doesn't really make sense to express this peptide, right? If it activates NK cells. So I think what CMV actually does, and that's why these peptide sequences actually overlap a lot with HLA-C, HLA-B, and HLA-A sequences. And that is kind of illustrated here because in a healthy cell, if HLA-E is expressed with self-peptides, I think one major function here is also to inhibit NK cells that express a very closely related receptor, which is CD94 NKG2A, this heterodimer. And what happens during CMV infection is that if HLA-A, B, and C are downregulated to evade CD8 responses, then these stabilizing peptides are being lost. That would lead to a loss of inhibition of this green subset of NK cells. That's why CMV, I think, on purpose actually mimics A, B, and C sequences to maintain this inhibition. And I think this deactivation through NKG2C is just another counterbalance from the immune system to fight back the immune evasion. That's why I think it's not surprising that CMV, because it has such a long evolutionary history with humans, that it has really adapted to express or to provide these HLA-C-like peptides in the absence of HLA-C. But then there are really some variants which do not are not identical to HLA-A molecules. And so I think that's coming back to the introduction where I touched upon, there will be always a balance between inhibition and activation, because for a healthy host cell, we do not see activation of NKG2C because that is overridden by other inhibitory receptors. So they usually do not lead to autoimmune responses because they have usually express NKG2C together with another inhibitory receptors, inhibitory KIRs, that are mostly specific for HLA-C and B, sometimes also for A. And then that gives them really this overdrive in the right situation here when CMV evades CD8 by downregulated A, B, and C, still providing E. And then these blue cells here, which express NKG2C and this inhibitory receptor, they lose inhibition and gain activation. And that, I think, is really what this response is about, to kind of fight back the immune evasion by cytomegalovirus. Yep.
Thank you. That's very interesting. Derek, you have a question?
So that was a beautiful talk, and I think it really convincingly shows that peptides bound to MHC class 1 molecules can influence the differentiation of NK cells. Do you know, could that be done by other molecules such as inhibitory KIRs? Or non-inhibitory KIRs. Would those peptides maybe influence NK differentiation?
So that's first of all, thank you for the nice words, and also, like, you're 100% on the right track. So there's one group from the UK where they have shown that actually a very, very specific substrain, I think a very, very rare isolate of CMV, can provide a peptide for HLA-C that then triggers an activating KIR. And what is, so that is, I think it was KIR2DS2, I want to say, but I'm not 100% sure. And the very, very interesting part is that the downstream signaling of activating KIRs and NKG2C through DAP12 is identical. So again, that could be that in a similar way with NKG2A as an inhibitory receptor, and then NKG2C as the kind of activating counterpart. This similar system can also happen with KIRs that are usually inhibitory and then can have some activating counterparts. And I think, like, really, if you think on an evolutionary basis, it could be that some of the gene duplications that have generated this many, many NK cell receptors really were maybe generated because of pressure to find a way to not be exploited. No, that's an excellent point. And I think it has been shown at least for one peptide, probably more.
Yeah, I think it's eventually coming to an explanation by why you can have one ligand can either activate or inhibit an NK cell depending on the receptor.
Yeah, absolutely. It will be very, very context specific. Yeah.
Thank you.
Great. Well, thank you very much indeed, Quirin, for your talk.